Q26.1. Describe the stages of oogenesis in human females. Don't miss a lesson, hit subscribe. Primary follicle stage. Oogenesis is initiated during the embryonic development stage when a couple of million gamete mother cells, oogonia, are formed within each fetal ovary. No more oogonia are formed and added after birth. These cells start division and enter into prophase I of the meiotic division and get temporarily arrested at that stage, called primary oocyte. Oocytes. Each primary oocyte then gets surrounded by a layer of granulosa cells and is called the primary follicle, figure 3.7. A large number of these follicles degenerate during the phase from birth to puberty. Therefore, at puberty only 60,000–80,000 primary follicles are left in each ovary. Secondary follicle stage. The primary follicles get surrounded by more layers of granulosa cells and a new theca and are called secondary follicles. Tertiary follicle stage. The secondary follicle soon transforms into a tertiary follicle, which is characterized by a fluid-filled cavity called antrum. The theca layer is organized into an inner theca interna and an outer theca externa. At this stage that the primary oocyte within the tertiary follicle grows in size and completes its first meiotic. It is an unequal division resulting in the formation of a large haploid secondary oocyte and a tiny first polar body. The secondary oocyte retains bulk of the nutrient-rich cytoplasm of the primary oocyte. Graffian follicle stage. The tertiary follicle further changes into the mature follicle or Graffian follicle. The secondary oocyte forms a new membrane called zona pellucida surrounding it. The Graffian follicle now ruptures to release the secondary oocyte, ovum, from the ovary by the process called ovulation. Q.80. Schematic diagram of oogenesis and spermatogenesis.